Good day, students. In the previous lecture, we have started about the single membrane bound cell organelles, and we have discussed the structure as well as the function of endoplasmic reticulum. In today's lecture, we are going to talk about the other single membrane bound organelles like Golgi complex or the Golgi apparatus. So we start with the Golgi apparatus. As I told you earlier also that the Golgi complex or the Golgi apparatus is a single membrane bound organelle. In fact, it is a structure made up of numerous membrane-like structures, flat sac-like structures that we'll be discussing later on also. So first of all, I must say that it is single membrane bound organelle just like endoplasmic reticulum we have discussed earlier. It is named Golgi complex or Golgi apparatus in the honor of the scientist who discovered them, that is Camillo Golgi, Golgi in 1898. So it was discovered by Camillo Golgi. In the plant cells, the Golgi complex or the Golgi apparatus is also known as dictyosomes. So in plants, regarding their presence, as we all know that the Golgi apparatus or the Golgi complex is present in the eukaryotic cells but absent in the prokaryotic cells. Also, in case of the eukaryotic cells, there are some type of cells which lack the Golgi complex, and the examples are red blood cells and the sieve cells. So we can say they are absent in the prokaryotic cells as well as in some of the eukaryotic cells like the red blood cells, also known as RBCs or the erythrocytes, and the sieve cells, which are the component of the complex tissue phloem in the plants, which is used, the phloem tissue is helpful in the conduction of organic nutrients. Remember that the red blood cells and the sieve cells are the examples of the eukaryotic cells. Even being eukaryotic, they lack the Golgi complexes. Now coming to the structure. Structurally, the Golgi complex is made up of numerous flat sac-like structures which are called as cisternae. And these cisternae are arranged in a stack one above the other. So we can say they are made up of flat sac-like structures. These cisternae are arranged one above the other in a stack. And these stacks of Golgi complexes are surrounded at the circumference above as well as below by the vesicles and the vacuoles, which are the rounded structures. The Golgi complex or the stack of cisternae takes a shape of a cup or the bowl. So we can say the Golgi body appearance wise looks like a cup or a bowl. If we draw the diagram, it looks like the flat sac-like structures represent the cisternae, which are arranged in a stack-like structure. And this stack of cisternae is surrounded at the circumference both above as well as below by the rounded structures which are known as the vesicles. As we can clearly see that the Golgi complex forms a cup or bowl-like structure which has two faces, the convex face, which is also known as the cis side, which is also known as the forming face, cis or forming face. It is the convex side of the Golgi apparatus, and it is generally this cis or the forming face lies near the nucleus or towards the nucleus. The other face of this Golgi apparatus is known as the trans or maturing phase or maturing phase. And this side is also known as the concave side due to its concave appearance. And this trans phase generally lies towards the plasma membrane of the cell. As we can clearly, one thing is more to observe in this diagram, that these flat sac-like structures which are known as cisternae are broader in diameter at the ends in comparison to the middle region. In the middle region they are narrower in diameter, but at the ends they are much wider in the diameter. Now coming to the functions of the Golgi apparatus. If we talk about the function of Golgi apparatus, their primary function is secretion. So they are secretory in nature. They help in secretion of polysaccharides, proteins, etc. So therefore they are present in large number in the secretory cells. Polysaccharides are also known as the carbohydrates. Besides secretion, they also help in the packaging as well as modification of the lipids and proteins. Also, they help in the cell plate formation and later on middle lamella formation in the plant cell. And this process occurs during cell division, that is cytokinesis step. Also, the Golgi apparatus helps in the synthesis of melanin, which is a pigment, and the hormones, other hormones. So it helps in synthesis of also it is the site for assembly of new membrane material. After the Golgi apparatus structure and function, we'll be discussing the next organelle, which is also single membrane bound organelle, and it is the vacuole. As told you earlier, the vacuoles are also the single membrane bound organelle, and the membrane of the vacuole is known as tonoplast. If we talk about its occurrence, it is absent in the prokaryotic cells but present in the eukaryotic cells. And on the basis of vacuoles, we can differentiate between the plant and the animal cells, because in animal cells, the vacuoles are smaller in size and they are numerous, whereas in the plant cells, they are few or just one, and they are quite large in size. In the plant cell, the vacuole can occupy up to 90% of the volume of the plant cell. Generally, these vacuoles arise from the Golgi complexes. Now, if we talk about their structure, these are single membrane bound spherical or oval rounded structures. Now, coming to the functions of the Golgi vacuoles. The main function of the vacuole is storage and transport of the various substances, and these substances can be nutrients or secretory or the excretory products. So they help in storage as well as transport of various substances. Besides this, the vacuoles are very important in maintaining intracellular turgor pressure in the plant cells. And this turgor pressure is essential for maintaining for making the plant cells intact, appear uh, in proper structure. Also, the vacuoles contain color pigments like anthocyanin, and that impart color to the parts of the plant. Besides this, some organisms contain a special type of vacuole which can change its shape and size, and it is bounded by a different type of membrane which can contract, and it is known as the contractile vacuole, and this contractile vacuole helps in the osmoregulation process. The osmoregulation can be defined as the maintenance of the water and salt balance in the cell. So, the contractile vacuole are specialized type of vacuole which are present in some of the organisms like amoeba, which is a protozoan, And this vacuole is bounded by a specialized elastic membrane which can help in changing the shape and size of the vacuole. And this vacuole is known as contractile vacuole, and it helps in the osmoregulation process, which is similar to the excretion process, which is related to in fact the excretion process. And osmoregulation can be defined as the maintenance of the water and salt balance in the cell. So this is all about the function of the vacuole. We'll be discussing about the other organelles in the next lecture. Good day to you.